Hi guys, we're gonna talk about genetic engineering and DNA manipulation. In genetic engineering, after knowing what's the DNA and genes, now we should know how can I use the genetics and how can I use the knowing of the nature of the genes and DNA in treating diseases in medical purposes, by inserting the DNA of human inside bacteria, to cut the DNA, to form DNA fingerprinting. So how can I cut the DNA and how can I deal with the DNA? Can you handle the DNA? Actually, you cannot. We cannot handle the DNA. So for DNA manipulation, we will need tools. What are those tools? Number one, I might use bacteria and bacterial enzymes. Bacteria and bacterial enzymes. After that, can I put all the data inside my mind, or I'll use computer programs? So I need computer programs. Well, we use chemicals to deal with the DNA, of course. Of course, in manipulating the DNA, we will need also chemicals. Those are the tools. Let's talk about cutting the DNA. Try to imagine that this is your DNA. So, to get a piece of DNA, you should cut. You should cut the DNA by scissor. Here is it. Can I use the same scissor in cutting the real DNA to get piece, to get only insulin, the gene for insulin, or the gene for any trait? No, of course not. I cannot use it because I cannot handle the DNA. I cannot even see the DNA by my naked eyes. So which scissors will we use? We will use enzymes from bacteria. Those enzymes are called restriction enzymes. Those enzymes are called restriction enzymes. So what's the function of restriction enzymes? To cut the DNA. What's the source of restriction enzymes? They discovered those restriction enzymes inside bacteria. Why do bacteria use restriction enzymes? To cut the DNA of viruses that invade bacteria. Like you have immune system, also bacteria use those restriction enzymes to kill invaders, to kill viruses. Well, restriction enzymes cut randomly the DNA at any pieces, at any site, or restriction enzymes have certain sites. Actually, each restriction enzyme has its specific site, and the site is called restriction site. One enzyme can cut at a site, let's say it's A, T, T, C, others C, G, C, G, C, A, and so on. So different enzymes have different restriction sites. The site composed of nucleotide sequence, and the nucleotide sequence ranges from 4 to 8 nucleotides. From 4 to 8 nucleotides. Okay. Do all restriction enzymes cut the same piece of DNA into the same number of fragments? No, they will cut so different enzymes cut at different sites and into different numbers of fragments. Okay. Some restriction enzymes cut the DNA into blunt ends. Look, blunt end. Those pieces have blunt ends, regular ends, like that. What if the DNA had this shape after cutting? This end is called sticky end. Because this sticky end composed of 3 nucleotides here. Let's say it's ATTA. So it's sticky because if I cut the DNA, any other DNA with the same restriction enzyme, having nucleotides complementary, they will stick together. They will stick together. So those are the sticky ends. Let's see how. If this is the DNA, and after cutting the DNA with the restriction enzyme that forms sticky end, this will be the shape. This end is called sticky because it has nucleotides that can pair with other nucleotide, A, T, T, C, G. So, if I cut another DNA, let's say it's human DNA, and I'll cut the bacterial DNA by the same restriction enzyme to have another sticky end, but complementary to it. T, A, G, C. So they will pair together. That's why those ends are called sticky ends. Those ends are called sticky ends. So there are 2 ways for restriction enzymes. Some restriction enzymes cut the DNA to form blunt, regular ends. Others, sticky ends. Others form sticky ends. And using restriction enzymes that form sticky ends is very helpful in joining 2 different DNA together because the sticky ends can stick to any other end that is complementary to it.